Hello, welcome again. Today we will be discussing about the enzymes, metabolism, role of these enzymes in the metabolism, along with the prosthetic group and the co-enzyme. And in the last, we will look at the various classes of the enzymes and how they participate. Some of the enzyme. We will take the example of some of the enzyme, how that participates in several type of the metabolic processes, metabolic reactions. So, starting uh, with this, uh, we have already covered the metabolism. Uh, Metabolism, we know that it's the combination of the catabolic processes and the anabolic processes. One is the degradation type of the reactions, and other is the biosynthesis type of the reaction. And normally this catabolism, that is also called exergonic type of the reaction, oxidation type of the reactions. And against that, the anabolic processes that consist of the androgenic type of the reactions, such as the biosynthesis processes, as well as the reduction type of the process that build up the high energy compounds into the cell. Both of these processes, anabolic and catabolic, that are active in the cells in all the living organisms. So we are talking about the human being. So these anabolic and the catabolic process that are active at all the time in human beings. Anabolic process, that require the energy, and catabolic process that releases the energy. And on the other hand, anabolic processes produce the more complex compounds, while the catabolic processes that convert the complex substances into the simpler type of the substances. So, with the enzymes, we have talked about in the previous lecture a lot more about the enzymes. So, some of the enzyme that need some cofactor, coenzyme, and prosthetic group. What are the differences among these terminologies? Cofactor, basically the that are actually the small molecules that are attached with the enzyme for the functioning of the enzymes. Um, and enzyme without its cofactor, uh, that that doesn't work sometimes. So the part of the proteinaceous part of the enzyme that without the cofactor, that is called as the apoenzymes, and the complete catalytically active enzyme is called as the holoenzyme. So apoenzyme, that is the protein portion, when the cofactor attached with it, that is technically called as the holoenzyme. So further, these cofactors that can be uh, further subdivided into two groups. It may be a metal type of the substances, or it may be a small organic molecules. So most of the common cofactors that are in the form of the metals ions, sometimes the ferric, sometimes the ferrous, sometimes copper, and some other metal ions. So cofactor normally refer to the inorganic type of the substances in this case. The cofactor sometimes that are organic molecules as well. If tightly bonds, the cofactor that will be called as the prosthetic group. If that are loosely bound, these cofactors uh, have the function of the coenzymes. So, prosthetic group means that are tightly integrated with the enzymes structure. Maybe it's a covalent bonding, it's a non-covalent type of the bonding, and organic type of the substances that can also bind with the enzymes. For example, pyridoxal phosphate, FMN, FAD, TPP, thymine pyrophosphate, biotin, These are the organic substance. You can see most of these are the vitamin from the water soluble vitamins, and in the form of the inorganic metal ions, copper, cobalt, magnesium, manganese, ferrous, lot more that can be used. So below this table that provided a brief about what kind of the metal ions that are combined with different type of the enzyme. For example, zinc carbon that is attached with the carbonic anhydrase. Sometimes zinc combined with the alcohol dehydrogenase. Sometimes it combined with the carboxypeptidase. Similarly, ferrous and the ferric cytochromes that are associated with the cytochromes. Cytochrome oxidase is potassium that is associated with the propanol CoA carboxylase. Magnesium that is associated with the hexokinase. This hexokinase that is especially important for the transfer of the phosphate groups. And when we talked about these coenzymes, uh, mostly these are the water-soluble water-soluble B vitamins, and its derivatives. For example, NAD, nicotine adenine dinucleotide, that is associated with the lactate dehydrogenase enzyme. NAD ग्लूटामेट dehydrogenase enzyme के साथ भी attach होते हैं. Flavin adenine dinucleotide that is associated with the monoamine oxidase coenzyme A acetyl-CoA carboxylase. TPP thymine pyrophosphate that is a derivative from the thymine. Vitamin B1 that is associated with the pyruvate dehydrogenase complex and that is the part of the glycolysis cycle, this pyruvate dehydrogenase complex. Then pyroxyl phosphate, that is the vitamin derivative from the B6, vitamin B6, and that functions with some of the enzyme that is associated with the amino acid metabolism. So we can see transaminases, decarboxylases, glycogen phosphorylases that work with this one. Biotin normally that is associated with the pyruvate carboxylase. So these are the example how these organic or the inorganic type of the molecule that are associated with the enzymes to work to help them work out. So this is again some of the example you have to see these example and how the coenzyme and the prosthetic group that is working with the enzyme. So now we will in this part, second part, we will see the classes of the enzymes. So enzymes that have basically 6 classes, and you have to learn these 6 classes in the same order: EC1, EC2, EC3, EC4. EC1 mean oxidoreductase. It's a big class. It's all of these are the big classes having different kind of the enzymes in it. So EC1 mean oxidoreductase, EC2 mean it's a transferase class, EC3 it's a hydrolases class, EC4 liases class, EC5 isomerase class, EC6 ligase class. So EC1, as the name indicate, oxidoreductase that consist of some of the enzymes because it's a big class. That is all of that enzyme that is associated with the oxidation and reduction process. And we have discussed the different definition of the oxidation and reduction in the first lecture in the class. So look at this one again, that it's a big class, consists of several subclasses. Oxidoreductase that also consist of, look at this table, dehydrogenases, oxidases, oxygenases, hydroxylases, or oxidases. So these are the subclasses that belongs to the EC1 oxidoreductase class. Oxidoreductase normally that is performing the function of oxidation and reduction reactions. All of the enzyme that is performing the function of oxidation and the reduction reaction. Addition are dehydrogenases enzymes that are associated with the Dehydrogenase that are associated with the addition or removal of the hydrogen. Oxidases, these accept two electron transfer. Oxygenase is important, that incorporate oxygen to the product. Similar is the case the second class transferases. The most important subclass is the kinases. Kinases basically, as the name indicates, all of these enzymes that are actually transferring some of the components from one substrate to another substrate. So kinases mean that is the kind of the enzyme class, subclass that is associated with the phosphate transfer. Wherever you see the kinase, it means there is a phosphate transfer through the enzyme. Then hydrolases. Hydrolases basically causes the hydrolysis process. Lases. You can see all of these classes that have the ending of the ases. So enzymes that have the ending of the ases. So hydro mean water. So hydrolysis of CO, CN, OP, and CS bonds. Esterases, these are the subclasses. Esterases, proteases, phosphatases, deamidizes. So all of that are the belonging to the class of the hydrolase. Lyase mean to break down. So addition across a CC double bond, and dehydrases, hydratase, decarboxylase that belongs to class of the lyase. Isomerase, as the name indicate, creation of the isomers. So there is an intermolecular rearrangement. Ligases, that is synthesis of the binding that belong to the function of all the enzyme is to synthesis. So formation of the bonds between the two substrate, that is the function. Now we will look in detail these about these classes and the nomenclature, how that can be used. So for example, you have a written EC 2.7.1.1. So this is the actually the nomenclature, how the one enzyme that is identified. This is the main class, this is the subclass, this is the subclass, this is a subclass. So if you are actually looking this 2.7.1, so this is the main class. This is the main class. The second class, you know, that is the EC2 mean transferases. So what does it mean? This is a transferase. So the 7 number, that is a subclass. What does it mean? This is a transfer of phosphate. This show the transferases. So 7 number mean that is referring to the enzyme that is associated with the transfer of the phosphate group. And next one is the subclass. So this is the subclass. This shows the transferase. This shows the transfer of the phosphate. This shows the phosphate where is going to transfer. So this one mean this is transferring the phosphate on an alcohol group, OH group. So OH is replaced with the phosphorus, and this one tells actually a specific name, ATP dehexo 6 phosphate transferase, and commonly that is known as hexokinase. So look at this one, 2.711, that we've seen in the previous slide. 2 mean it's a class. In this case, 2 means this is a class of the transferase. This is the class of the transferase. So 7 mean it's a subclass, transfer of phosphate. This one mean it's a subclass, alcohol is the phosphate acceptor, and this one mean ATP exose deposits, specific name. So all the enzyme nomenclature that is written in this form, EC 2.7.1.1 or 1.2.1.1. So This is the, if that is starting with the 1, it's mean it's an oxidoreductive. If that is starting with the 2, it's mean it's a transferase, and so on. This is the main class. So look at this with the same enzyme, uh, hexokinase. Hexokinase, as we have discussed here, that is actually referring to the phosphate transfer that belong to the transferase class, and that refer to the phosphate transfer to the alcohol. Alcohol mean OH. Alcohol mean OH. So look at this one. Uh, this is the hexokinase. Hexokinase means transfer of the phosphate that belong to the class of the transferase. So you can see uh, this alcohol group. So phosphorus is actually going to transfer at that point. This is the alcohol group. This is the alcohol group, and the phosphorus is going to transfer to that. It's mean from where this phosphorus is coming. That is coming from the ATP. So this phosphorus is transferring to the OH alcohol group, and hexokinase that is transferring this phosphorus to this one. So we can see here it's a transferase. What it going to transfer? Transfer the phosphate group. Where it going to transfer that? To a alcohol, right? So look at this one. Hexokinase is belonging to the class of the transferase. What it is transferring? A phosphorus. Where it is transferring? To an alcohol group. So look at this one. So I think you are getting these points and how that enzyme that is going to work. And this process I have taken from the glycolysis process, and you are well aware of the glycolysis process. This is the glycolysis taken from the initial point of the glycolysis process. So these are some of the other example. You have to see all of these. The first class is the oxidoreductase, second class is the transferase, third is the hydrolase. I have given some of the one or two example from different pathways. So from the oxidoreductase, alcohol dehydrogenases. This is a subclass, dehydrogenases of the oxidoreductase. So we can see ethanol that react with the NAD, nicotine adenine, and this is a coenzyme that is working with this alcohol dehydrogenase, as we have seen. This is an organic type of the substance. So that is doing the process of the oxidation and reduction. In the original form, this is in the reduced form. This ethanol is in the reduced form due to the presence of this OH, and this is in the oxidized form. So as I told you, alcohol dehydrogenase is an enzyme that belongs to the oxidoreductase class. So there is a, what we are expecting, there is an oxidation process, there is an reduction process. So after, what is the function of this one? And we know that this is in the reduced form, this is in the oxidized form. So after the function of this one, that is going to reduced, and that is going to oxidized. So look at this one, acid. This is forming the acetaldehyde. So it's a now oxidized, and this is reduced. This is actually reduced. This is the reduced form. So we can see both of the function oxidation reduction is happening by alcohol dehydrogenase. Transferase, we have discussed in the previous slide how the transfer take place, right? Another example, this is the example from the protein, urea cycle, transaminases, alanine, and alpha ketoglutarate, that is forming carbon skeleton of the pyruvate, and glutamate in transaminase, that is belonging to the class of the transferase that is active. Another example is the hydrolysis. So several subclasses for the hydrolysis. One is the subclass is the peptidase, and hydrolysis mean breakdown in the presence of the moisture. So we are looking at this one, breakdown in the presence of the moisture. So look at this one. This is basically amino acids. So that is producing a shorter type of the it's a bigger type of the polypeptide molecule. In this case, that is composed of the two amino acids. So that is breaking this peptide. It's a dipeptide, so that is breaking this dipeptide in the presence of the moisture. Lyase is that is involved in the addition of a group to a double bond, or removal of a group from a double bond without the hydrolysis, uh, oxidation. And these are the subclasses for these one. So pyruvate that is converted into the acetaldehyde by the pyruvate decarboxylase, releasing the carbon dioxide. Isomerase mean to form the isomers. And there are several isomerase, malate isomerase that is converting the malic acid of the malate into the fumarate of the fumaric acid. Ligases that is bonding of molecule using the ATP. So energy is required as a synthesis reaction. So pyruvate that is using the ATP, the pyruvate carboxylase that is converting it into the oxaloacetate. So that's all for today, and you have to look all the classes of the enzyme and uh, what are the basically examples uh, of these enzymes. You must know the several enzymes that is working into the different reactions. So we will meet in the next lecture, inshallah.